Boston, and I'm a professor of medicine and ophthalmology at Boston University School of Medicine, and I've been working in this field for more than 30 years, and so I've been very closely associated with diabetic retinopathy and the complications that come from ocular diseases, especially from the retina. My area is in the area of diabetic retinopathy.
Tell me about the United Scientific Group EYE 2019 meeting.
EYE 2019 is the first meeting that has been organized by the USG, or the United Scientific Group, and this meeting focuses on bringing together talented scientists who are experts in their field and try to have a kind of a homely environment where you can interact with each other in a closed setting. Otherwise, what happens is when you go to these big meetings, they're lovely and you have all the topics you want to discuss over there. But because it's so big, there's an inherent issue with that. You can't find certain experts to talk to, or you have parallel talks going on everywhere. You want to go to this one, also you want to go to the other talk, and that sometimes doesn't happen because it's running in parallel. So big meetings are great, and I attend those big meetings, but sometimes you also like to have smaller meetings where your interactions are much more easier in terms of accessibility.
How often are they planning on having this meeting?
You know, so far I am getting this feedback that they would like to have it every year, and I have to talk to the organizers and find that out.
After the meeting, is there a way for participants to follow up or continue conversations about the research presented here?
There are a number of things going on. There will be a website. We have an app that allows us to be in touch with each other, whether you want to ask a question or you just want to have a contact email or phone number. So we have that kind of a app that's also tracking all the recent changes, any updates in the final program. Those are all available through that app. So that's one way. The other is to connect to the USG website, and over there will be again excerpts from the meeting in small, abstracts will be accessible. So if someone wants to know a little more in detail, it's not like a small snippet of the title of the talk, but a little bit more, that's also possible.
Can you tell me about the research you presented here?
Sure. I work on, as I said, diabetic retinopathy, which is the leading cause of blindness in the working age population. And unfortunately, there is no cure. So we need to work hard to find a way to prevent this retinal vascular lesions that develop during diabetic retinopathy. And my work focuses on understanding structural damages that happen to the small capillaries that are present in the retina. Especially cells start to die and disappear, and also the vessels start to become leaky. So our focus is to understand what are the molecular mechanisms underlying this kind of breakdown and dysfunction of the capillaries, which lead to blindness and vision loss.
Are you simply looking for the cause, or are you also working on a therapeutic at the same time?
We are working both. We are trying to understand the cause, because if we could identify the steps, the molecular pathways, factors that are involved in the pathogenesis of diabetic retinopathy, then we can start targeting those pathways. If a gene is being overexpressed, then we would try to go in there and try to bring it down and normalize that, and thereby hope that the functionality is maintained. With respect to translational research, we are working on a specific molecular approaches using antisense oligonucleotides or siRNAs, small RNAs and DNAs, but these are tools that allow us to go and fix genes that are abnormally expressing themselves in this disease. So we are close to two of those. One, I gave a talk on day one here, and that relates to basement membrane thickening in the retinal capillaries. And what happens is when the retina starts to dysfunction, we have been able to observe that the basement membrane thickening can promote vascular cell loss and leakage. Other people who are working in this field have also found similar findings. And now the question is, can you fix that? And we have moved to the next level, and we have shown that in an animal model of diabetes, you can actually, because the animal models express the same kind of pathological signs. For example, they also have leakage and they also have basement membrane thickening in their capillaries in the retina. So if you perform intravitreal injections using antisense oligonucleotides and siRNAs, you can actually normalize the abnormal gene upregulation and thereby prevent vascular leakage in the retina, or fix the basement membrane thickness. You can bring it down to normal levels. So we have now shown that in an animal model. Our next goal is to see if this works at the human level. So this is one example.